Okay, so we're just finishing off the last part of the lesson that we didn't finish yesterday. Yesterday we had been talking about multiple alleles. We talked about blood type. We talked about blood type and some other ideas. So for example, blood type and male or female. So we looked at those two Punnett squares. We talked about having an order or a hierarchy of dominance. So we had the bunnies yesterday or the cows today with their coat color. Another scenario that we could consider is something called epistatic interactions. Epistasis is the phenomenon wherein the effect of one gene is dependent on the presence of one or more modifier genes. So this is the first time we're really considering that one gene affects another gene. Up until this point, genes had been separate from each other. They could be inherited together if they were linked, but the two linked genes didn't necessarily have an impact on each other. In this scenario, genes are having an impact on each other. And this is probably what a lot of real life scenarios are actually like. A lot of times people have a gene, but might not end up expressing it the same way as the next person because of another gene they have that modifies the action of the first. So remember, a gene is a section of a chromosome that codes for a protein. And the way the protein functions or whether the protein is even produced could be affected by the protein that gets made by another gene. So we're going to look at an example with rooster combs. That's the hair feathers they have on the tops of their heads. In this example, there are two different genes that are on two different chromosomes that work together to impact the shape and style of the rooster's comb. So in this example, we have the P and the R genes. Now they're on different chromosomes, which means independent assortment is still happening. But once we see which assortment we have for both of those genes, then they affect each other. So they're still inherited independently, but once we see the full genotype, they work together to create the phenotype. So if I consider the P allele, capital P, if the rooster has the capital P allele, but it doesn't have the dominant R allele, it will have a P-shaped comb. So if I look over here, this second one on the top, that's a P-shaped comb. If the rooster has just the dominant R allele, but doesn't have a copy of the dominant P allele, it will have a rose-shaped comb. That's that kind of fancy one down there at the bottom. If the rooster has both dominant alleles, instead of having a P and rose shape, it'll have a different shape called a walnut shape. And the walnut is this very pompous looking one over there on the bottom left. And if the rooster happens to have neither of them, it has just recessive alleles, it'll end up with a single comb, which I think is like the classic rooster comb that I imagine when I imagine what they look like. There are other possibilities, but this is the simplified version. So you can see there were other shapes there that didn't come up because they required different combinations. So we're going to imagine that in a parental generation, we crossed organisms who were pure breeding, and we ended up with an F1 generation where all of them were heterozygous. Okay? So we're imagining that's how we got to the F1 generation. What'll really be interesting is to look at what happens when we make an F2 generation, where we take two organisms from the F1 generation and cross them. Okay? So this was our F1. We are going to make an F2 generation out of that. Now, hopefully you look and you're like, hey, we did this kind of a cross before. This is the kind of cross we did when we looked at dihybrid crosses, when we looked at Mendel considering two traits at a time. The only thing that's different here is that instead of considering two traits, this is only one trait, one phenotype, but two genes are controlling it. So it'll look like a dihybrid cross, but we're only going to get one trait out of it. So let's set up a Punnett square. Here's mine. To make the F2 generation, I'm going to cross these two heterozygous genotypes. If you look at my Punnett square, you'll notice that each parent could make 4 different gametes. a gamete that had both dominant alleles, dominant R, recessive P, recessive R, dominant P, and both recessive alleles. So you look at this and you say, yeah, this looks the same. We did this. This is a dihybrid cross, except that instead of having two traits like tall and green, or short and yellow, we're only considering the shape of the comb because the two genes work together. Now in this particular scenario with rooster combs, the way these genes interact, we end up getting a very similar looking scenario to our dihybrid cross, but again, we're just considering one trait and not two. So all around the edges of my Punnett square, when I mix those two gametes together, the outcome that I get is at least one copy of the dominant allele for both genes. You'll notice in the first column and first row, the genotypes are not all identical, but they all give me the same phenotype. All of these combinations that give me at least one copy of both dominant alleles give me the walnut comb shape. And if you count, there are 9 of them. If I consider the rose shape, you get the rose shape if you have dominant R, but you do not have the dominant P allele. So here, and down at the bottom, even though they're not all the same genotype, they all give me the same phenotype. I have at least one copy of the dominant R allele, and I do not have the dominant P allele, so I end up with a rose phenotype. These 3 right here, kind of in the bottom right corner, all give me the P phenotype. I do not have a copy of the dominant R allele, but I do have at least one copy of the dominant P allele. And we said that that is how I get the P comb shape. And down at the very bottom right, just like in a dihybrid cross, I get one possibility where the organism is homozygous recessive for both genes. And in this scenario, it gave me a single comb shape. My ratio was the same ratio that I got in my dihybrid cross when I crossed 2 heterozygous organisms, but I was considering only one trait instead of 2. So this is an example where, just like with intermediate and codominance, our Punnett squares didn't look different, but the way we interpreted the outcome was different. The way we interpreted the phenotypes was different. Now, if you have a question you would like to ask about this example, I would love to know what it is. So I'll just pause for a second here because I can see people are writing. Now, you do not have to memorize how rooster comb shapes work. That would apply to any scenario we talk about. You don't have to memorize the details of the scenarios. That's what the contexts of the questions are for. So I am recording this in case you ever need to go back and take a peek at something, but I do want to try to finish the last slide here. There are other traits that we're not going to consider in terms of Punnett squares because they start to get a bit too complicated. Most of the traits that we have considered are what we would call discrete. They have like 1 or 2 options, maybe 3 options for the phenotypes, but there are a lot of traits that we would call continuous. Maybe you remember learning about this in grade 9 science. I remember I used to teach it. It's like the very first thing I would teach in this unit. But you have a lot of characteristics, and so do other organisms, that are what we call polygenic, where poly, which means many genes affect that trait. So continuous traits are ones that have a very gradual variation from one extreme to the other. A really good example is size of really anything. If we look at the traits we've considered so far, a lot of them are either you have it or you don't. If we looked at Mendel's crosses, the plants were either tall or short. But if we consider people, there are way more than 2 choices. There's not tall and short and that's it, and those are the only 2 heights people can be. There's tall, super tall, really tall, very, very, very extra, extra tall, medium short, a little bit shorter, really short, super short. There are lots and lots and lots of variations. And so size is a trait that we would usually consider to be continuous. There are other traits like that. There are traits like the color of things, especially like the color of skin, for example, would be a polygenic trait, where it's not like you have either very, very white or very, very black skin. There are a million possibilities and variations in between the extremes. So we can't classify these continuous traits like we could with colorblind and not colorblind, because they depend on more than one gene. When there are lots of genes that contribute, the ratio of phenotypes becomes a gradual curve. And so depending on where you are in math yet, have you guys learned about bell curves in math yet? I think you learned about that in Math 30. I don't actually know though. Do they learn that? Maybe. But what it means is instead of having 3:1 as a ratio, or 9:3 to 3:1, we would have a ratio that's like 1:2 to 4 to 7 to 10 to 11 to 50 to 30 to 12 to 10. There would be so many different possibilities that our ratio, if we mapped it out on a curve, would give us more of a curve shape than anything. Now we are going to run out of time. That's okay.